Hello, everybody, and welcome to our bacterial identification presentation, The Mystery of the Water Bottle Bacteria. The location we swabbed was Andrea's water bottle on both sides and the straw. This was done to test just how important washing reusable water bottles is. Throughout this experiment, we isolated and identified an unknown bacterium from this environmental sample. For our hypothesis, we expected to identify Bacillus subtilis from the environmental sample. It's a common soil and water bacterium that creates a sticky coat and makes it easy to transfer to many fomites. Because of this, the bacteria could easily be on our swabbed water bottle. There are many strains, so it's a very versatile bacterium. We performed a whole bunch of different tests. Some of them were helpful, such as the gram stain, endospore stain, starch hydrolysis, and RNA sequencing tests, which we will go over more in a minute. And other tests that we ran did not help us identify our bacterial isolate. Those were the catalase, citrase, and sodium broth tests. When grown on a TSA plate, the unknown colony appeared a yellow-white color, formed large colonies, and had low bait margins, a flat elevation, and a shiny surface, as depicted in the picture above. The bacteria under a microscope was rod-shaped, indicating that the unknown bacteria could belong to the Bacillus genus. It had what appeared to be endospores in the center of some of the bacteria. It didn't appear to be arranged in any particular type of grouping. Single, bacterium, diploid, multiple, and branch-like groupings were all visible. In the gram stains, it showed both positive and negative gram bacteria, indicating possible gram variability. The cell diameter was also found to be smaller than 1 micrometer. The endospore stain test ended up being positive, as we had predicted. Endospores were located in the center of the bacterial cells and were ovular in shape. The endospore-containing cells were observed not to be swollen. This narrowed our choices to either Bacillus or Clostridium genuses. It was reasonable to conclude that the bacteria was not a strict anaerobe, since it was isolated on a TSA plate. Bacillus can be assumed. The positive control for the endospore stain was Bacillus subtilis, and the negative control was E. coli.
Next, we performed the starch hydrolysis test. The starch hydrolysis test is used to identify bacteria that can hydrolyze starch using the enzyme amylase. The ability to hydrolyze starch is depicted by the halo present around B. cereus, the positive control bacterial culture, as shown on the left side of the image. The negative control, E. coli, on the right, and our unknown on the bottom of the image, show visible growth, but no apparent halo. While our bacteria was able to grow on the starch agar, no amylase was produced, giving us a negative result. In the microbiology lab at CWI, we isolated 16S rDNA from our unknown bacteria and sent it to TACGen for sequencing. Original DNA sequencing returned 0 results due to an unknown error in the in-lab DNA isolation procedure. In March, due to SARS-CoV-2, aka the coronavirus pandemic, classes were moved to being completely online and the campus was closed. This made running additional tests or DNA isolation not possible for us. Thanks to our amazing professor, Teresa Rich, our unknown bacteria was restreaked onto TSA plates and sent directly to TACGen for genetic analysis. DNA from the unknown bacteria was extracted and amplified using polymerase chain reaction. The DNA from the forward and reverse strands were sequenced. We viewed and edited both forward and reverse DNA sequences using the computer application codon-code aligner. The beginning of both sequences can be seen here in this image. The highlighted green nucleotides were deleted because the sequence was not amplified in sufficient quantity. Using the NIH library of known bacterial species DNA sequences, we were able to compare the forward and reverse sequences and locate species that showed up in both searches. Two bacteria were found to be 99.5 to 100% identical in both the forward and reverse, Bacillus C and Bacillus pumilus. We looked into each of these bacteria to determine which of these species matched the closest to our unknown environmental bacteria based on the gram stain, colony morphology, endospore placement within the cell, and where it can be found in the environment. So, to review, using the Berge Manual of Determinative Bacteriology flowchart, the following test results were observed. The gram stain test revealed gram-positive rods. The endospore stain revealed central spores and spores. The fact that the environmental bacteria was isolated from a TSA plate meant that it was not an obligate anaerobe. A negative starch hydrolysis test disproved our original hypothesis of B. subtilis, but the flowchart led to indicating that if the BP test was positive, the unknown bacteria could have been positively identified. Additional tests were run, but were unnecessary. The 16S rDNA sequence from TACGen was analysis confirmed that the starch hydrolysis test and BT test would identify the proper bacteria. Bacillus pumilus. Our original hypothesis that it could be B. subtilis proved to be close. The cellular features of B. pumilus are synonymous with B. subtilis. Also, when looking at the phylogenetic tree of Bacillus species, you can see that these two are very closely related. B. pumilus matches our experimental findings. On this slide, you can see images of known Bacillus pumilus. The colony morphology is similar to what we observed and reported. It is gram-positive, rod-shaped spore-forming bacteria. It is found in soil, water, and other habitats. It is not known for being pathogenic in humans, so it's not going to cause me any issues if it's on my water bottle. There was a lot of different bacteria on my water bottle. If they're all as friendly as B. pumilus, it doesn't seem that additional washing of my water bottle is necessary to maintain my health, but it's probably a good idea. B. pumilus is significant to the ecosystem by transforming molecular nitrogen, N2, into ammonia, NH3, which plants love. Now let's wrap it up. Since B. pumilus is normally found in both soil and water, it is reasonable to think that it could be present on my water bottle. Our line of thinking that led to our original hypothesis that it could be B. subtilis was solid, since they are so closely related. Now on to issues we had this semester. We misread Berkey's flowchart and did not realize that B. pumilus was an option if we had just run the VP test and gotten a positive result. So we ran more tests than necessary. We also had a very difficult time growing B. pumilus in broth. The original DNA sequencing returned 0 results due to an unknown error in the in-lab DNA isolation procedure. The coronavirus pandemic added new challenges to a microbiology lab learning experience, but thanks to a problem-solving professor, we were able to complete this bacterial identification project. Here are some images from our additional yet unnecessary tests. Thank you for watching our presentation.
We hope this presentation has changed your life and encouraged you to wash your water bottles and containers more frequently, just in case.
It has been an interesting semester, but we learned a lot and enjoyed getting to know all of you lovely people.